Ellie Abbott, and this is my research project, the effect of fatty acid structural isomers on the rate of conjugation in E. coli K12. Antibiotic resistance is scary, and we should all be way more scared than we are about it. Currently, 700,000 people a year die of antibiotic-resistant infections around the world. But according to scientists, that number could increase to as many as 10 million deaths a year worldwide by the year 2050. Now, for comparison, the current leading cause of death is cancer. That kills 8 million people a year. So antibiotic resistance spreads in two ways. The first is called natural selection. So there's always a small proportion of resistant bacteria in any bacterial population, which I've shown here in pink. Now, every time we use antibiotics, we kill off the susceptible bacteria, shown here in gray. That allows the resistant bacteria to multiply and take over the population. Now, the other way that antibiotic resistance spreads is called conjugation. So in this case, bacteria actually share resistance genes with one another, which turns those susceptible bacteria into resistant ones. This isn't the main way that antibiotic resistance spreads, but it has been proven to be a significant contributor to the spread. Now, natural selection is a basic function of biology. It's basically evolution in miniature. So antibiotic resistance is inevitable to some degree as long as we use antibiotics. The spread of antibiotic resistance is definitely being accelerated by human activities that increase the exposure of bacterial populations to antibiotics. Those include overprescription, misuse of antibiotics due to poverty, and the use of antibiotics in agriculture. So current solutions have tried to address natural selection by reducing these factors, the goal being that would reduce exposure to antibiotics, reduce selective pressure in favor of resistant bacteria, and ultimately reduce the amount of antibiotic-resistant bacteria in the population. But these problems are huge and global. There's so many interconnected factors. Policy in one country is not going to solve antibiotic resistance worldwide. So research has turned to biochemical methods to slow the spread of antibiotic resistance. Now, as I said, natural selection is pretty fundamental and pretty hard to mess with. So a lot of this research has focused on conjugation. Now, in theory, if a chemical could be found that inhibits conjugation, we could prevent the sharing of antibiotic resistance genes and ultimately slow the spread of antibiotic resistance. This chemical could maybe be administered alongside an antibiotic to prevent conjugation from happening in the body. Now, even if this works perfectly, it will not stop antibiotic resistance. It's not going to solve this problem, but it could slow it down enough that it buys us time to continually develop new antibiotics, which is the only way to really combat this problem over the long term. One molecule that's been identified as a conjugation inhibitor is a fatty acid called 2-hexadecanoic acid, or 2-HDA. Researchers have tested various characteristics of this molecule to see what makes it the most potent. They've found that a 16-carbon chain, a carboxylic group, and an unsaturation, or triple bond, are necessary components for maximum potency. But one characteristic they have not tested is the location of that triple bond. So I wanted to do just that. Investigate if the location of the triple bond impacts the potency of this molecule as a conjugation inhibitor. To do that, I investigated the potency of a molecule called 15-hexadecanoic acid, or 15-HDA, which is identical to 2-HDA in every way but the location of the unsaturation. So to test this, I used 2 strains of bacteria, each of which were resistant to one antibiotic. I combined them in a liquid culture along with various concentrations of 15-HDA and allowed them to conjugate, or not. This produced bacteria that were resistant to both antibiotics, which I've shown here in green. Then, I plated this whole liquid culture onto a petri dish containing both antibiotics. And I incubated that overnight. So the unconjugated bacteria were resistant to one antibiotic but susceptible to the other one. So they died. And all I had remaining were bacteria that were recombinant, the result of successful conjugation, and they were resistant to both antibiotics. So each of those recombinant bacteria replicated to create a colony. And this is what it looked like in the actual petri dish. So each one of these circles, each one of these dots, is a colony that started from a single recombinant bacterium. So I collected 2 kinds of data from this. The first was the actual number of colonies. For this Petri dish in particular, it was 160. Now, in theory, that is again the exact number of recombinant cells, so it should be the best measure of how much conjugation occurred. But it's impossible to count on some of these Petri dishes. The colonies were just too dense, too close together. So I calculated a second measure, percent cover. That's just the percent of the area of the Petri dish that's covered by colonies. So I did this using Photoshop, and for this Petri dish, for example, it was 8.2%. So when on Petri dishes where I could measure both of these values, I ran a linear regression test and found that they were highly correlated with an R-squared of 0.95, which suggests that percent cover is a reasonable proxy for the number of recombinant cells. So I performed this in triplicate at each of 5 concentrations. And for each Petri dish, I calculated both of those two data points as a percent change from the control, which of course had no 15-HDA. Now, before I get to the main data, let me take just a minute to discuss an important control in this experiment. You see, it's possible that 15-HDA could have actively killed or harmed all of the bacteria, rather than specifically prevent conjugation. To ensure that this wasn't the case, I performed the same experiment, but rather than plating onto a Petri dish that contained both antibiotics, I plated onto a Petri dish that contained no antibiotics. This allowed me to see if the concentration of 15-HDA had any effect on the number of all bacteria, rather than just the recombinant ones. That's this data here, and you can see from the error bars that there's no significant difference in the total number of colonies, which suggests that 15-HDA does not have any generalized effects. That means that we can assume that the effects that I show you next are due to 15-HDA specifically inhibiting conjugation. So on the left here, we have a graph of the number of recombinant colonies. And on the right here, we have a graph of the percent cover. Now remember that both of these are presented as a percent change from the control. You can see really similar trends in both graphs. They make the same shape. Now, I'd just like to point out here that the reason that there's no bar here is not because it was the exact same as the control, but because all 3 Petri dishes at that concentration had colonies that were just too dense for me to count, so I wasn't able to collect data for this measure. But at all concentrations, both of these 2 measures displayed the same direction of change in comparison to the control, and at least one of the measures was significantly different from the control. Now, let's look at the trends here and compare them to the data from 2-HDA. At all concentrations of 2-HDA, which included these concentrations and more, the amount of conjugation decreased from the control. So we see that here at the higher and lower concentrations. There was a decrease in the amount of conjugation that occurred compared to the control. But the really interesting thing is that here, at the middle concentrations, we actually saw an increase in the amount of conjugation. That's not known to occur at any concentration of 2-HDA, and that's fascinating. Now, because of this unexpected data, I could not calculate a potency measure for 15-HDA that's directly comparable to the published data for 2-HDA. But I think it's safe to say that there's a clear difference between the conjugation inhibitory properties of 2-HDA and 15-HDA, which means that I can conclude that the location of the unsaturation in hexadecanoic acid does indeed affect the potency of the chemical as a conjugation inhibitor. So while 15-HDA is probably not a great candidate for production as a conjugation inhibitor, there are many other isomers of this molecule. And now that we know that each of them could display different potencies, it's possible that some could be more potent than 2-HDA. Clearly, we need a lot of future research to explore this possibility. And again, the ultimate goal here is that one of these could be administered alongside antibiotics and be used to prevent conjugation and slow the spread of antibiotic resistance. Thank you very much.